Okay, children, good morning, everybody. Let's talk about the fundamental unit of life today, that is cell. So, today we are going to learn about cell, and you know that all living organisms, whether we, plants, animals, you talk about any living organism, is made up of cell, right? So, it was first of all, there are a few terms of scientists that you need to remember. Robert Hooke was a scientist who was actually working with thin slice of cork, and he was the one who actually coined the term cell, right? So according to him, cell means tiny compartments. If you take a peel of onion bulb and you just try to take the concave side of it and peel an onion, and you would see, if you take a thin layer of it and stain it with saffron, you would see tiny compartments in it. Again, those compartments are nothing but cells. So that is what he meant when he talked about cells. Then second one, that is Leeuwenhoek, another scientist. Now he was the one who actually used an improved microscope, and he actually discovered living cells in pond water. So at his time the technology got improved, so he got a chance to actually work with an improved microscope, and he was able to find living cells in pond water. Next we need to learn about Robert Brown. Robert Brown discovered nucleus inside a cell. So in some books you see they say that nucleus is considered as the brain of the cell, and basically it actually stores the genetic information. So Robert Brown was the one who discovered nucleus in a cell. These names are important, that is why I'm summarizing it initially. Next we come to another scientist by the name Purkinje. Right? Now this scientist is known to coin the term protoplasm. So he coined the term protoplasm, which is the living fluid part of the cell. So protoplasm was coined by Purkinje. Next there were two persons, Schleiden and Schwann. Now they were the ones who proposed the cell theory. So they proposed the cell theory, they gave the cell theory according to which all living organisms came into being because of the cells. So cells are the structural and functional unit of life. This was proposed by Schleiden and Schwann. Later on, there was this scientist by the name Virchow, and he kind of elaborated the cell theory and added to it that all cells are made of pre-existing cells. All cells are made of pre-existing cells. So life cannot exist if a cell is not there. New cells arise from pre-existing cells. He added to that. Now, if you talk about organisms based on the number of cells, then we basically have unicellular and we have multicellular organisms. Now, as the name suggests, unicellular organisms will have single cell. Some of the examples would include amoeba, paramecium. Then you have unicellular alga by the name Chlamydomonas. So we have Chlamydomonas and bacteria. All these are unicellular. And on the other hand, we, plants, animals, fungi, all these are multicellular, right? Okay. Now every cell, if you talk about every cell has got a basic structure you take the case of any animal cell, it will have the outermost membrane known as the cell membrane or the plasma membrane. Then it would have a fluid filled inside, the fluid matrix known as cytoplasm, and nucleus, the control center of the cell, right, that contains the hereditary material. So every cell has got this. If you talk about plant cell, then they have an additional wall besides the cell membrane, which is absent in animal cell. Now let's talk about the cell membrane first of all. Now this plasma membrane or the cell membrane is supposed to be selectively permeable, right? It's obviously the outermost covering of the cell. Outermost covering, you can say, it gives identity to the cell. It separates one cell from the other, and it permits the entry and exit of certain materials. So we can say this membrane is selectively permeable. That means not all the things can cross it. So there's a kind of check imposed by this membrane. Now there are a few terms that you need to understand so that you understand how the membrane functions. One is osmosis and another is diffusion. Now remember, whenever we are talking about the concentration of water molecules, right, we are referring to osmosis here. So when we talk about movement of water molecules happening from, not concentration, let me write movement. So when we talk about movement of water molecules from a region of high to low concentration across a semi-permeable membrane, that is referred to as osmosis, right? For example, if you talk about raisins, when you soak raisins in a bowl, the raisins are obviously going to imbibe water and they are going to swell up. So the outermost membrane of raisin is the plasma membrane or the cell membrane, and you see the water gets inside because there is a difference in concentration of water molecules across the membrane. So if you are taking the water molecule concentration, if you look into that, inside the bowl you have high concentration of water molecules, and inside the raisins the molecules of water are less concentrated. So there is net movement of water from the bowl into the raisins, and which is why raisins swell up. So that is dealing with movement of water molecules. So remember, osmosis deals with movement of water molecules from where? From high to low concentration across a semi-permeable membrane. Now if you are talking about diffusion, we are talking about movement of solute molecules. Again, we are talking in terms of solute concentration. So when we are talking about solute molecules, again from a region of high to low concentration across there is no membrane here, so from a high concentrated region to a low concentration region, so that is referred to as diffusion. For instance, if you are lighting an incense stick in a room, obviously the fragrance of incense stick is more concentrated where the incense stick is burning. So the fragrance is going to travel from that region, from where the incense stick is burning, to the other parts of the room, right? So that is what is referred to as diffusion. Now if you talk about, again, suppose there let's study different cases of osmosis. Let's say we have a plant cell, right? I have this plant cell or animal cell. Let me just take any random cell, and when we are putting it in sugar solution or salt solution, right? So solute is going to be more in case of sugar solution or salt solution, right? So what is going to happen? We can say that, let's say this is the cell, right? And you have high water concentration, high water around the cell. Let me take this case first. We have high water molecules, or the concentration of water molecules is high around the cell. Now this is the case of dilute solution. Obviously solute is less, solvent is more, right? So what is going to happen? The water molecules are going to move from a region of high concentration to low concentration, which is why the cell is going to gain water. Cell gains water by osmosis and swells up, right? This is the first case. Let's talk about second case. Now let's say you have the same cell, and let's say it has the same water concentration outside as well as inside the cell. Let's say the medium has exact water concentration as in the cell, right? So what is going to happen? Obviously, if the thing is balanced, then no net movement is going to take place. So neither the no net movement of water, right? So the cell is going to be of the same size. Cell remains of the same size. That was the second case where you have same concentration. Now let's see a scenario, a case where you are putting the cell in a medium which has low water concentration outside. So low water concentration in the medium, and inside the cell the concentration of water is more, or you can say the cell is very turgid or swollen, right? Now you have put it inside low water concentration. Now what is going to happen? The cell is going to lose water, because again the water molecules are going to move out from a region of high to low concentration, right? So it's going to be referred to as, the cell is going to lose water and the cell is going to become flaccid. Okay, now such a solution, if you talk about the types of solution, then that solution that has high water in the medium, such a solution is referred to as hypotonic solution. Obviously the solution of solute is less than that of water. Here this is isotonic solution, which has same concentration both outside and inside, and this one is referred to as hypertonic solution. That is that solution in which there is low water concentration in the medium around the cell, right? So these terms are, you should understand how exactly osmosis works. Now because of this, we say that plasma membrane is basically flexible. If you talk about characteristics of plasma membrane is a flexible membrane. It is semi-permeable membrane, and it is going to allow certain things which are going to get in and get out of it, right? If you talk about its structure, it's made of lipids and proteins, and it helps to take the material from the external environment, right? And this helps the membrane to do endocytosis. That means taking in something, right? It's because osmosis helps it to take in the things, or diffusion helps it to take in or give out the things through it. Okay, now let's talk about cell wall. Now as I said, cell wall is present only in plants, not in animal cells, right? So they have additional membrane. This is an additional membrane besides the cell membrane. So they have cell membrane. If you look at a plant cell, they have cell membrane, then they have additional membrane around it. Now this membrane is what is referred to as the cell wall. Now this is made up of cellulose, and so it provides the structural strength. It provides structural strength for the plant to withstand any kind of environmental, you can say, winds or whatever tries to move the plant. So they can just keep standing and resist any kind of movement. So it gives structural strength to the plant to stand all day and all night. So when plant cells loses water by osmosis, let's say the osmosis takes place and the cell loses water. Now what happens to this? The cell wall remains as such, and the contents of the cell are going to shrink, because the cell is obviously going to shrink in size, right, away from the cell wall. Such a cell is referred to as plasmolized cell. Plasmolized cell. That means the cell contents have shrunk because of loss of water due to osmosis, right? So this is important. Plasmolysis is the process and cell is said to be plasmolized. Okay, now let's talk about nucleus. Now nucleus, as I said, we say it's the control center of the cell. Now it's basically a double layered structure. It has two membranes, right? And if we talk about the outer membrane, it has pores on the outer membrane. Obviously the things have to move out of it, get inside it, right? So it has to have pores. Then there are, since nucleus is dealing with heredity, so it has chromosomes, which are the genetic material contained inside the nucleus, right? And these chromosomes are made up of DNA and histone proteins that make up the chromosomes. Now this chromosomes has got short segments of DNA called genes, and these are functional parts of the chromosome, right? Now as long as the cell is not dividing, this chromosome is contained in the form of chromatin material, which are nothing but thread-like structures which are present inside the nucleus. Now if this is the nucleus, as long as the cell is not dividing, the chromosomes are going to be highly coiled like this and present like the chromatin material, right? When the cell is supposed to divide, then this is going to condense and it is going to take the normal form of chromosomes like this, right? So they become chromosomes when a cell has to undergo cell division. Okay. Now, one very important role of nucleus is that it helps in reproduction, or you can say division of cell and growth of an organism, right? Now, on the basis of structural complexity, we have basically two types of organisms, right? One set of organisms are referred to as prokaryotes, and the other ones referred to as eukaryotes. Now, what are the differences? Now, one thing that should come into your mind is that if you talk about prokaryotes, they have simple organization, or you can say they are primitive forms, and they have more complex organization inside the cell. Complex organization. Now, how is it complex and how is it simple? In case of prokaryotes, if this is the cell, the genetic material, which is referred to as nucleoid, lies suspended inside the cytoplasm. So there is no nuclear membrane around the genetic material, right? And it is also referred to as nucleoid. Also, there is lack of well-developed organelles, right? Now, if we talk about the eukaryotes have got complex organization. They have well-defined nucleus. Now, they will have properly defined nucleus with proper nuclear membrane and chromatin material lying suspended. Now, this is the nuclear membrane, which is not present in case of eukaryotes. And also it has many different organelles like mitochondria, lysosomes, other organelles, ribosomes, endoplasmic reticulum. So this is much more evolved. Now this is present, eukaryotes would include all the plants, animals, fungi. All these are eukaryotes. Whereas prokaryotes, we just have bacteria, bacterial cell, and blue-green algae. And these are the examples of prokaryotes, right? All right. I hope this thing is clear. Now let's move on to the next part, that is cytoplasm. Now, as I said earlier, cytoplasm is the fluid component of the cell. Cytoplasm is the fluid matrix or the fluid component in which all organelles are suspended, right? All organelles are suspended. They are just floating around in that medium, and it is a site of many chemical reactions. Okay, now if we talk about organelles, you have endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, all that. Let's talk about the first organelle, that is endoplasmic reticulum. Now, endoplasmic reticulum is like a network. There's a network formed by this, right? It's actually it's made of membrane-bound tubules, or you can say sheets, right? And they have similar structure as that of plasma membrane. Now, if it is rough endoplasmic reticulum, that means it has ribosomes attached to it. Okay, so if you see it under the microscope, you would see that it appears quite rough. The surface doesn't appear to be smooth because it has ribosomes attached to it, and ribosomes are the site of protein synthesis. If we talk about smooth endoplasmic reticulum, it has no ribosomes attached to it, right? So ribosomes would be lying suspended around in the cytoplasm, but they are not attached to the endoplasmic reticulum. Now, ribosomes, as I said, they help in protein synthesis. You can say they are the protein factories of the cell. They are the protein factories where proteins are made. Now, why do we need proteins? The proteins are needed for manufacturing the cell membrane and various other organelles and all that, right? Okay, now these proteins are sent in cells through the endoplasmic reticulum. Fine. Now, there is the smooth endoplasmic reticulum that also helps to make fats and lipids, and some of which is used to make the cell membrane. And some lipids function as enzymes or hormones, and basically the job of endoplasmic reticulum, if we have to summarize in a nutshell, it is to transport proteins, whether inside the cytoplasm or between cytoplasm and the nucleus. In case of smooth endoplasmic reticulum, this is basically involved when the liver has to be detoxified. Let's say a person has had enough of alcohol, right? And the effect of alcohol has to be detoxified from the liver. So here comes the role of smooth endoplasmic reticulum. Now we move on to the next organelle, that is the Golgi apparatus. Now this Golgi apparatus is also referred to as Golgi bodies. So Golgi apparatus, and this was basically described by a person by the name Camillo Golgi, and hence the name Golgi apparatus. Now these are nothing but these are flat sacs of membrane-bound vesicles, and they are arranged in stacks. Let's say these are the stacks, parallel stacks like this, right, called cisternae. They are arranged in stacks like this, called cisternae. And what is the function? Functions include storage, right? They store certain things, modification, packaging, repackaging of products. Okay, so they are like factories, packaging products before they are sent out, right? So for example, if you talk about simple sugars, the simple sugars can be assembled to synthesize complex sugars. So that's the job of Golgi bodies. They are also involved in the formation of another organelle called as lysosomes. Involved in formation. They are involved in formation of lysosomes. Right? Let's look at the function of lysosomes now. I'll try to brief it up quickly. Now lysosomes are also, they are again, first thing, membrane bound sac. They are having membrane around it. They are filled with digestive enzymes, right? And they are made by rough endoplasmic reticulum. Now they are also referred to as the waste disposal system of the cell, or sometimes they are referred to as suicide bag of the cell, because if the cell loses the function due to any reason or goes out of order, then the lysosomes enzymes are going to digest its own cell and leading to its death, right? So the function, you can say, is to digest worn out organelles. All those organelles which are not functioning well, it's going to digest them up and kill them up. Next we come to mitochondria, the powerhouse of the cell. I believe you would have read about it in your class 7. Firstly, they are, okay, let me write, they are referred to as powerhouse. Why powerhouse? Powerhouse of the cell, because they generate something called as ATP, adenosine triphosphate, which is referred to as the energy currency of the cell, and it is involved in every activity of cell. It powers the cell. Now this is double membrane structure. Double membrane has double membrane. Then outer membrane is porous, and very important process of respiration takes place here, occurs here, to produce ATP. Now this body is going to use ATP for making new chemical compounds and also for mechanical work, right? Now one thing more, that they have their own DNA and their own ribosomes, right? Okay, next we move on to plastids. Now plastids are only present in plants. Plastids are not present in animals, so only in plant cells. And basically we have two types of plastids. So we have chromoplasts and we have leucoplasts. Now leucoplasts are involved in storage of various things like oil, protein, fats, starch, all these things, right? And chromoplast, if they are the colored plastids, right? The flowers get their colors due to chromoplast, and if chlorophyll is present in it, then it is referred to as chloroplast, right? And like mitochondria, plastids also have their own DNA and ribosomes. Now they are made of membrane layers called thylakoids, and there's this liquid called stroma, right? So that constitutes the plastids, the chloroplasts basically. Let's move on to vacuoles, the next organelle, vacuoles. Now these vacuoles basically are storage organs, you can say. They're the storage sacs. Now anything solid or liquid can be stored in it. These vacuoles are small in animal cells, and a huge central vacuole is present in plant cells, right? Now what do they store? They store many amino acids, then they store sap, organic acids, proteins, etc., right? Okay, a cell is going to possess structure and specific function because of its organelles, and that is why we say it is the fundamental structural unit of living organisms. Now we move on to the last topic, that is cell division. Now as the name suggests, the cell has got the ability to divide, and because of which new cells replace the old worn out cells or dead cells, right, or injured cells. Now there are two methods of cell division. One is mitosis, another is meiosis. Now in case of mitosis, basically most cells divide by this method. Most cells divide by mitosis, and it leads to growth of the organism because all the cells are similar and they all have same number of chromosomes as the mother cell. So same number of chromosomes. Mostly all the cells divide by mitosis. Whereas if we talk about meiosis, now here there are specific cells of reproduction. There are specific cells of reproductive organs, you can say, or tissues in animals and plants, and they divide to form, these all divide and they form gametes. Divide to form gametes, right, by meiosis. Now these gametes after fertilization are going to produce offsprings, but what is important here is that two consecutive divisions occur in meiosis, two consecutive divisions, whereas here we have only one division happening, in this case mitosis. Now because of this division, you have 4 new cells produced by a single cell, right? And another thing, these cells have half the number of chromosome than parent cells, right? Now whereas here you have same number of chromosomes as the parent cell. So I hope this topic is clear to you. Children, happy learning. Just go through the notes and revise it again. See you all again in the next video.